Welcome to the Germline Gene Polymorphism Day for the AIR community webinar series. And as usually, we'll have a established researcher speak for an hour, and then we'll after an hour, then we'll have an early career person. This time we start with Melissa Smith from the University of Louisville, and after an hour, we'll have Oscar Rodriguez. So, Melissa, I think you're ready. So take it away, please.
Okay, super.
Oh, excuse me.
Oh,
and
yeah.
I was told I have to push the AIR Community 7th International Meeting. It will be in Porto. I can assure you that'll be a great time. June 3rd through June 6th, and registration is live, and Pam will put the registration in the chat, and we all encourage you to. It'll be a great time, great science, and great discussion with colleagues. So now, please, Melissa.
All right. I'm glad I didn't get you into trouble by starting too quickly. So thank you so much for the invitation. I'm really excited to share some of the work we've been doing here at the University of Louisville, looking at the genomic foundations of immune diversity and function, which is a slightly different title than I gave Pam a few weeks ago, but we'll wrap back up to autoimmunity at the end. So the basis for my interest, at least, in learning more about immunogenomics is to understand why vaccines, although sometimes incredibly robust, are never 100% effective. So we all know on this call that response rates vary from 30% with seasonal flu vaccines to over 90%, most recently with the SARS-CoV-2 vaccine. But understanding what factors underlie this differential responsiveness is limited and is usually tagged to poor immunogen selection or adjuvant influence. And this has led to the idea that we can possibly develop, what is quote unquote called a universal vaccine, sort of a one shot to rule them all type of approach. This is exemplified by the measles, mumps, and rubella vaccine that's given here in the US. So 90% of children receive the MMR vaccine every year. That's over 3 million newborns, but up to 10% of these children won't respond. So that's over 300,000 children a year. As a parent, this is somewhat terrifying. Up until the discovery of SARS-CoV-2, measles was the most infectious virus known to man, but only 20% of that response variation is currently understood and attributed to adjuvant differences or viral strain differences. So recent work from our group and the group of Corey Watson has suggested that genetic background influences immune response potency. I'm sure to the majority of the people on the call, I'm preaching to the choir, but I do want to go over a little bit of that evidence to convince you why we're laying the foundation for further work looking at these variants in the repertoire. So as all of you know, antibodies have myriad varied effector functions. What we're very interested in our lab isn't the business end of the molecule or the variable region that dictates antigen specificity, but really which functions are dictated by the FC region. I personally believe that this region has suffered from being misnamed as the constant region. Of course, it's constant compared to the variable region, but as I hope to convince you today, there is lots of polymorphism in the constant region that really needs to be further understood. And it comes from germline variation in the Ig loci. So as everyone here again knows, usually I introduce this slide by not wanting to trigger anyone's PTSD, but I think all of us are a little bit of gluttons for punishment here. The human immunoglobulin locus is highly polymorphic and complex. And while this may be the view that we get it in our typical immunology textbook, the VD, J, and constant region cassettes are far more extensive and complex than that's what's typically taught, right? With a number of variable genes, diversity genes, joining genes, and pseudogenes that make up the variable region area that recombines into that VDJ gene cassette. And then, of course, of our interest, the constant region genes down here outlined in light blue. So working with Corey Watson here, pictured with our fancy new PacBio Revio instrument, we've developed two novel targeted methods, both of which I'll touch on lightly today. IGenotyper, which I'm sure you've heard about from Corey in the past. I know Oscar will probably talk about this quite a bit as well after me. And then the FLAIRR-seq method, which basically takes the idea of looking at a comprehensive picture of the germline repertoire to see if germline variation impacts the circulating RNA antibody transcript repertoire, the express repertoire, and how these variants impact expression at that level. And of course, our overarching hypothesis is that there are baseline foundational differences in the germline sequences that encode both Ig and TCR that impact which antibodies or T cell receptors are capable of being expressed in the repertoire, and therefore sort of obviate the traditional hypothesis that we all start on a level playing ground in terms of response to infection, vaccination, and cancer, that your ability to respond to different pathogenic insult is going to be dictated by what is contained in your germline. So I want to start a little bit with IGenotyper because that's laid the foundation of where we've gone since then. So Corey and I have been working on this assay for, I guess, now more than 9 years. And it's based on a targeted method of capturing very specific long-range DNA fragments. So at this point, we're up to sort of 10 to 12 kb fragments with an oligonucleotide pool specific for our locus of interest. Today, we'll be talking about Ig, but we also do this with TCR. And then we read this out in a long read sequencer. Today, we use the PacBio Revio system that we obtained here in August. The advantage of a targeted assay, of course, is it's relatively high throughput compared to whole genome sequencing, and it's less expensive than whole genome sequencing because we can multiplex many samples onto the same run. And of course, a long read sequencing can more effectively interrogate those repetitive V, D, J, and pseudogene cassettes that I showed you in the schematic previously. Those cassettes are 300 to 400 base pairs long. So if you use short read sequencing, you can often get mismapping and trouble assembling this locus. But if you're getting 12,000 base pairs, you can phase those cassettes in the proper order and overlap those reads to assemble with much higher accuracy. With the Revio system and improvements in sequencing technology and chemistry over the past several years, the average accuracy of these reads are now Q42 or over 99.99%. So this is also an order of magnitude more accurate than what you see with standard short read sequencing, just improving our ability to assemble these loci in the germline assay. So we published a paper actually written by Oscar, who's presenting after me, that was published last year in Nature Communications. I should probably fix that slide here. Really showing the haplotype variation in the variable region genes in particular. And so I want to just walk through this slide to make a point of how diverse this is, right? All of us are taught initially in Immunology 101 that we all have this VDJ locus. It's very complex. There's lots of VD and J genes, but that we all essentially have the same genes, and that the random mix and matching, I can feel Corey twitching from here, the random mixing and matching of those genes is what creates differential response. So I would argue that what we've shown over the past 3 or 4 years with our publications and what our lab is very focused on now, labs are very focused on now, is that it is not at all random and it is not at all the same. There is no level playing field. We all have very discrete patterns of genetic variation at this locus, and that's what this paper showed. So this was the largest cohort to date, 154 healthy donors across a number of ancestral backgrounds, looking at the presence, absence, and variation within the variable genes, diversity genes, and joining genes. What was really striking out of the gate once these loci were assembled was the number and extent of structural variation within this locus, so here denoted by SVs, and those are represented deletions here by these dotted lines. So you can see that there are certain individuals that have giant pieces of their locus completely deleted. So if, for example, you wanted to target one of these genes with a therapeutic or vaccine strategy and people did not carry them because there was a deletion, they would have no chance of responding. There's a lot of other details in this figure, including insertions represented here by this triangle, but I'm not going to go into detail because it's an incredibly robust data set that would take the whole hour to explain. I think the other thing that was really striking, in addition to large structural variation, was that there was a lot of allelic variation as well. So we all know that there's allelic variation and copy number variation in this locus, but in these 154 individuals, Oscar was able to identify 127 novel coding alleles represented here, where the genes that have novel coding alleles are identified with the height of the bar representing the number of novel alleles that were found. And of course, a lot of those novel alleles were enriched of areas of high variation that we know in V169, in 330, but we're still discovering things every time we sequence someone, which is pretty astonishing. Something that I'm personally interested in, won't talk about it much today, but I'd love to chat with any of you about it later, is the pattern also of genes that are not polymorphic. There's genes that are not deleted and don't have any polymorphism in them across our individuals. And that's also a question on the other side of what are those genes doing and why are they so conserved? I do just want to highlight that these patterns of structural variation existed across individuals of all ancestral backgrounds. In this graph, we just represent the amount of the diploid DNA deleted from sort of the maximum IG locus possible, and the amount of that DNA deleted across individuals color-coded here by their ancestral background. This individual in particular had over half a megabase of their locus deleted, which comprised 30% of the locus and 25% of the coding genes. So, right, this person is walking around and labeled healthy, right? We bought these samples from Stem Cell Technologies. They're, ostensibly healthy individuals. So it's sort of amazing how much variation someone can withstand and still maintain at least a superficial healthy phenotype. So a lot of times I talk about this with people who are used to doing AIRR-seq profiling at the RNA level, and they say, well, so why should I really care what's happening at the DNA level? We really care about what gets translated into the RNA level. And so I want to talk a little bit about how these variations impact the antibody repertoire. So we took these data, the germline haplotypes, we cataloged the copy number variation and structural variations, as well as the SNP variation that we found across these 154 individuals. And then Oscar performed an eQTL analysis to identify which variants affected how the antibody genes themselves were affected in the repertoire. Obviously, in the case of this blue V gene, if it's gone, deleted, it's not going to be in the repertoire. But in some cases, there were non-coding variations here that did affect the ability of certain gene cassettes to be expressed in the repertoire. And this is where our technology really shines. A lot of people impute germlines back based on RNA sequencing or AIRR-seq data, and they miss this non-coding variation. And we found many, many SNPs in the non-coding regions that actually did impact the usage of V genes in the repertoire. And this is why we think that this DNA-based genotyping assay is really critical for us to understand repertoire dynamics going forward. So again, what does this really matter, right? Again, if you're reading your Janeway textbook and thinking about sort of your ideal immunology scenario, there's this idea that if there's one specific B cell floating around in your big toe and you have an antigen exposure in your cervical lymph node, that somehow those two will meet and mount a robust immune response. I would personally question that. I don't know of empirical evidence that really supports that sort of one cell is all you need model. And so we looked at this in the context of some infection and vaccination models to understand what the potential impact is of this variation in actually infectious insults. So the place that we started was with HIV vaccines. And the reason that we did that is because this is an area where people are specifically targeting B cell genes, specific V genes in the germline to elicit a very specific antibody response. So in the context of HIV, which is where I come from scientifically, only about 20% of infected individuals will raise natural, broadly neutralizing antibodies. And so there's been a huge effort over the last 30 years to identify those individuals, study their B cells, isolate those broadly neutralizing antibodies, and find out what makes them special. One of these antibodies is called VRC01, and there's a number of immunogens that have been designed to target the elicitation of that antibody. The specific chain that makes up VRC01 is this V12 gene, particularly the STAR02 allele. And what I mean by that is if you're homozygous here for the V12 STAR02 allele, you have an incredibly high percentage of your circulating B cells that use that allele, ready to respond to that immunogen or that antigen. To the contrary, if you express the STAR05 allele in a homozygous fashion, you actually express very little to negligible numbers of B cells that express V1-2. In fact, we couldn't even represent it as a percentage here on this axis because there were only 3 clones found, so sub 1% values. So if one cell meets one antigen and that's sufficient, maybe that magic can happen here. But if you think about sort of the way germinal centers are made and B cells are stimulated, the fewer numbers you have of a responsive clone should at least intuitively impact your ability to mount an immune response. And so just in case there's anyone that's unclear, I want to define what I mean by allelic variance. So in this case, we have 2 alleles. The main allele, this major allele, STAR02, that's the responsive allele. We're going to be talking a bit about the STAR04 allele here and the STAR05 allele. In both cases, they differ by a single residue, and in both cases, it's actually funnily enough, an arginine and tryptophan that are swapped from the major allele, the STAR02 allele. And if you actually look at B cells carrying V1-2 as a function of which allele they carry, these STAR04 individuals have a lower usage frequency of 1-2, as I just showed you with the STAR05 allele as well. So there is questions, right? 5-fold circulating less V1-2, that's the chain we need to hit to get vaccine response here. It is highly likely to impact vaccine responsiveness if you're just thinking needle haystack type of mentality. So this was published by Shane Crotty's group in collaboration with Corey in a Nature Vaccine paper in 2021. And actually a paper just a few weeks ago came out from Bill Sheaf's lab, oops, I'm going the wrong direction, looking again more at this genotype function, specifically the STAR05 and STAR04 alleles. So in this cohort, they had a number of participants here represented in this bar graph, had a number of different genotypes of the V1-2 gene, right? Several STAR04 alleles, and then a few that were heterozygous for STAR05. When they looked at the expression of this clonotype in the repertoire, they actually saw this amazingly correlated almost dose response curve of the frequency of the expression of this gene as a function of their genotype. So these plus symbols represent those individuals that carried a STAR05 allele, even just as a heterozygote, whereas the purple circles up here represent those STAR02 allele carrying individuals. So based on the allele you have, even in a heterozygote fashion, you actually have a dose response essentially of the presence of the reactive antibody in your circulation. So that's not promising, right? You have very low levels of the reactive antibody here. But if the antigen was able to meet that B cell, if you did get the one B cell from your big toe into the lymph node in your neck to meet that antigen, would it react? And to address that, this group did a BIA-core assay where they actually mutated the STAR02 tryptophan to an arginine to see if in the context of the STAR allele, if you only change that residue, what happens to the binding of antigen? In the STAR02, STAR04 context, they got good binding of the immunogen, the vaccine immunogen, at levels that were expected. However, when they mutated just that tryptophan to an arginine, this binding was not at all robust and pretty much at the top of the curve in terms of what they could measure. So the conclusions of this paper were really striking. So while not precluding that there could be an impact of these B cells in vaccine response, the fact that both precursor naive B cell frequencies were so much lower, and that had the likelihood of impacting effective responses, as well as the actual antigen binding affinity was much, much, much worse in these alleles. So leading to the idea that even if you could find the right B cell, it may not actually respond robustly, demonstrates the importance of these germline variation, how they act in the repertoire. So with that, I'm going to shift. So basically, based on these data, we wanted to see how not only variation in the variable region, which is what we're looking at here, impacts repertoires, but also that variation in the constant region. So I'm going to pivot to the other assay we developed, FLAIRR-seq here, and talk a little bit about some exciting data we've been seeing with that assay. So if we look at the RNA space, right, not only is the DNA structure complex, but the RNA structure is also complex. You have the recombined variable region containing the V, D, and J segments. And then with IgG, at least, you have 3 constant region exons. There are sequence motifs in CH1 that determine the isotype and the subisotype signatures. And we know, based on decades of literature, that it is this constant region, again, that dictates a lot of those effector functions that I highlighted at the beginning of the talk. Allelic level definition of the constant region is not possible with any of the current technologies, and I'm going to show you why. So current AIR-seq technologies are limited in our view. So you've got the very small amplicons generated for just clonotype identification using CDR3. There are primer pool-based methods to get the entire variable region. You have to know what you're looking for there. So, and then you have the technical challenges of a complex primer set pool and multiplex PCR. And then you also have the VDJ race approach that a lot of us use for AIR-seq, where you prime out of the constant region exon 1, you use a template switch oligo at the front 5' end so that you can capture novel alleles in your mix. None of these 3 methods provide much resolution of the constant region. So we developed, and the other limitation, of course, is that they're all limited by short read sequencing. So they're all sequenced either on the MiSeq or now the NextSeq 2000 or NovaSeq. 600 cycle kit, 2x300 reads, has that limitation of about 580 base pairs to ensure that you have enough overlap to assemble those reads. If you have extra long CDR3s, due to somatic hypermutation, you actually can exceed this length and not be able to assemble those reads. And so the read length constriction of that platform itself restricts what we can see in these repertoires. So with all of these sort of limitations, we went ahead to try to develop a full-length immunoglobulin transcriptome AIRR-seq profiling assay, and we call this FLAIRR-seq. So again, we start with the 5' race approach, but instead of priming in the constant region exon 1, we prime in the constant region exon 3. And we incorporate a barcode into that primer so that we can multiplex our amplified products downstream. The final length of these products for IgG is about 1.5 kb and IgM is about 2 kb. And then we sequence them on a PacBio sequencer. So the data that was in the paper, our JI paper last year, was mostly all generated on the Sequel IIe. As I mentioned, we're using the Revio now. The amazing thing about this technology is it doesn't have a length restriction, and the more times you can sequence that molecule in one well, the higher the accuracy gets. So we actually filter our data on Q40. All the data we look at is above 99.99% accurate, but the average read quality of these amplicons after coming off a PacBio run is actually Q60. So that's, again, 3 orders of magnitude higher accuracy than a lot of the competing sequencing technologies. So in our proof of concept paper, we took 10 individuals, healthy donors from Stem Cell Technologies. 8 of them were Caucasians. I think we had 1 Asian American and 1 African American, just as a proof of concept for FLAIRR-seq's capabilities. The first thing we did is we did IGenotyper, that first assay that I described to you. So we established ground truth of the haplotypes contained in these individuals using DNA. And then we took the RNA co-extracted from the same samples and ran our FLAIRR-seq assay. These reads were mapped to HG38. We actually trimmed the VDJ off in order to see if we could genotype the constant region just using the FLAIRR-seq assay. And then we compared to ground truth. I'm going to show you a few of these results now. So this is what the data look like. I actually find this quite striking. So this is the reads aligned in IGV. So the variable region is here, not surprising. It's a gamish of color. But then the constant region exons are here in gray, introns in blue. This is an IgG4, so there is a single hinge region. But you can see, although yes, more constant than the variable region, there still are polymorphisms across the constant region that can define these into allele-specific groupings. This allows us to phase these alleles and generate allele-specific clonotypes, allele-specific repertoire profiles. What's also interesting to us is there's a bunch of RNA biology in here that we don't think a lot of people have looked at. There's retained introns, there's deleted exons. I think there's a lot of functional genomics that we can do with this data to see sort of how malleable and plastic these molecules are while retaining the ability to get translated and turned into productive protein. We haven't touched that quite yet, but we're moving in that direction. Another thing to consider, right, is so there's a clonotype right here where you can see the expanded multiple reads with the same VD and J pattern of base usage. Now, with our technology, you can see the entire constant region. You can see if there are deletions in those exons or hinge regions, something that may affect downstream function that you wouldn't see with any other technology. When we look to see the genotyping capacity of this technology, this is what we saw. So let me explain this so that the different individuals that we assess, their codes are here on the y-axis. Across the x-axis are the different constant region genes specifically for IgG and IgM. Those were the chains that we tested. For anything that has a dot, we were able to generate a genotype with FLAIRR-seq that agreed to 100% with IGenotyper. Our primer in IgG1 is a little upstream of a SNP that determines the difference between 2 IgG1 alleles. So you can take it from the 20 or 30 that we know down to 2, but can't always get a definitive answer in IgG1 at this time, although we're hoping to improve on that in future iterations. What was incredibly striking to us is that across the 10 individuals, we identified 32 IgG and IgM alleles. 87% of them, 28 of them were novel compared to IMGT. Again, I'm sure that's not a super surprise to many of you on the call, but we were shocked at how much diversity there was that had not yet been explored. In particular, not only did we identify new alleles, so any allele here that has an FL in its name is novel. If it includes a star and a digit before that, it means that part of that allele is an IMGT, but we extended it through the rest of the constant region. So we just completed alleles that had been partially filed, essentially. In IgG4 in particular, we found 4 individuals, so 40% of our individuals actually had a duplication in one of their haplotypes of IgG4, leading to 3 alleles being used in the repertoire, not just 2. So this actually has become a theme in the lab, this identification of novel constant region duplications. And this IgG4 duplication, which you can tell in those individuals that have 3 boxes in their IgG4 column rather than the standard 2 alleles, is actually quite frequent in the populations we've been looking at. Very intriguing for IgG4 specific function, not something I have time to talk about today, but believe me, I could talk to a wall about it. I find it fascinating. So let's talk about what this means in terms of how you can analyze repertoires. So this is sort of where we're at with other AIRR-seq technologies, right? The number of unique VDJs per chain. So we have, over 15,000 unique VDJs in IgM, and over 6,000 in the sample in IgG. Now with FLAIRR-seq, we can break this down on a per sub isotype level for IgG and even a sub isotype allele level, which is incredibly powerful. So one of the reasons that we really were excited about doing this is we have a lot of interest in autoimmunity, which I'll get to in just a second. And there's many autoimmunities that are known to be IgG3, IgG4 mediated. Because IgG1 and IgG2 are so dominant in the circulation, and there was no prior way to segregate out just the IgG3 specific reservoir, you can get a lot of noise in trying to understand gene patterns and possible pathogenic alleles, autoantibody alleles, when you're looking at the entire repertoire. With our technology, now you can break it down and you can look at just the V, D, and J chains that are used per constant region allele. So let me show you what this looks like. So these are chord plots. This is the IgM data that's represented here above. We have both IgM alleles here on the top arc. So the green, say, is the maternal allele, and the purple is the paternal allele. Across the bottom, we have the IGHV families. So what we're looking at here with the chords are the genes from each family that are used per allele. And so we can then say, okay, well, this allele, this purple allele, the paternal allele, actually uses IGHV5 family genes in its repertoire, 2 more the same or less than the other allele. In the case of looking at IgG, you can break this down further by subisotype. So this top arc is far more complicated because it includes all the subisotypes of IgG. But what you can see is how much we can focus in away from the noise. So if you're really interested in looking at an IgG3 phenotype, you can just look at the chords that stem from these 2 arc fragments and ignore the repertoire data from the rest of these arc fragments, which really does focus your ability to interrogate subisotype specific repertoires in diseases where that pathogenicity is already determined. We also looked at how we could use this technology to track antibody responses over time. So this is just a proof of concept. We're making no biological conclusions, but we used a whole blood RNA isolation from 4 time points post-hospitalization for an individual that was hospitalized for severe COVID-19 in New York in April 2020. And then we looked at the relative proportion of the subisotypes within those individuals at those time points. So we have no idea when this person got infected. This is post-hospitalization. So you do see a shift in the different repertoire components. So far more IgG3 than you see at later time points. And if you actually identify single clonotypes or single clones in the mix, you can actually track them over time as well. So this is a clone that we identified. We're calling it 9900. So it has the exact same VDJ and CD3 signature. And over the time points, we can see it as a clone within the different compartments, IgM, IGHG1, and IGHG2 in the repertoire. And the shapes are color-coded by their time point. And what we can see here in this top square that's highlighted is actually class switch recombination happening in sort of semi-real time in this individual. So initially, this clone was part of the IgM repertoire. It shifted and underwent class switch recombination to become an IgG1, and then with later time points shifted even further to the IgG2 compartment where it appears to have stayed for the time points that we visualize. So if you're trying to understand antibody dynamics over time, particularly how clones are evolving longitudinally in a system, this is a great assay with which to do that. So once we proved that it worked and got our paper out, which was great, the next thing we needed to do was actually apply it to a biological question. So in collaboration with Kevin O'Connor at Yale and Steve Kleinstein at Yale, we decided to look into myasthenia gravis. And the reason that we wanted to look into myasthenia gravis is because first, it is known to be mediated by IgG1 and IgG3 autoantibodies. Secondarily, it actually has three distinct mechanisms, two of which are variable region focused, so internalization after antibodies bind and cross-link receptors, so that's going to be variable region determined, and a functional block mechanism here, again, binding the target of the acetylcholine receptor and blocking acetylcholine from engagement. But it also has an FC-mediated mechanism, and that's the fixation of complement and then destruction of the acetylcholine receptor. And there is a lack of association between titers, autoantibody titers, and response to therapy, autoantibody titers, and disease severity. So we had this idea that if we could isolate individuals that were using predominantly complement as their mechanism of autoimmunity versus these others, that maybe we could better subset these patients to know who maybe should be receiving complement inhibition therapy that would have the most effectiveness in those individuals. So the first thing we did was work with this pilot cohort, and this is the data I'm going to show you today, although we're vastly expanding it right now as I speak. So we started with actually 3 different B-cell autoimmune diseases, the acetylcholine myasthenia gravis, this NMOSD, and MOGAD, as well as healthy donors, 4 additional to the 10 that I just showed you from the FLAIRR proof of concept paper. We did IGenotyper and FLAIRR-seq in the matched fashion, as I mentioned earlier. So again, these are our individuals here on the y-axis, the IgG genotypes across the y-axis. You can see immediately there are actually 4 individuals here that have that IgG4 duplication that's schematized here. And in these 18 individuals, we identified 16 novel alleles. So really, anytime we're profiling a group of people, we're identifying something new, which is really powerful, and again, underscores how little we understand about the constant region. So our current efforts are really focused on annotating this constant region functional signature to characterize antibody activity. So let me tell you what I mean by that. So, we think about the antibody in its protein form, which is not something a lot of us AIRR-seq people do frequently. There have been a number of papers published by Maggie Ackerman and many, many, many antibody engineering groups that have identified specific residues in the constant region important for effector function. This particular review is amazing if anyone is interested, but they've identified many residues in FC that dictate the ability of IG to activate complement, whether that's binding C1q directly or affecting the ability of FC-FC interaction. So new to me, as I discovered, is that FCs actually hexamerize, and that increases their affinity for C1q. And so these residues here affect the ability of FC receptors to hexamerize. We've also identified this as a very small list of a very long table that we've put together. And namely, I want to call out Easton Ford, who I know is on the call. He's the graduate student that has led all these efforts. And it's through his very hard work that we have seen so many exciting results. So we've collated a table of known coding variation in the FC region that affects function. And we're starting to annotate these in our alleles of our myasthenia gravis individuals. So here's an example. So this is just a multi-sequence alignment of the IgG3 alleles, so CH2 and CH3. These are the alleles that are in IMGT. These 5 alleles are novel that we've identified in some of our data, although it's not exhaustive at this point. But for example, this alanine in just IG3*15 is associated with FC interaction, enhanced complement activity. Similarly, this tyrosine has been identified to enhance FC-FC interaction in about a third of the discovered alleles. What's really fascinating that I saw as I was sort of looking at this and wrapping my head around it, this lysine right here, lysine right here, actually knocks out an N-link glycosylation site. So this is an NXST motif. We haven't found anything in the literature, but I would anticipate that this probably has a big effect on FC receptor function just because of what we know about glycosylation of FC and function. And then something that we really found that was fascinating was actually the full deletion of hinge regions in IgG3. So canonically, IgG3 has 4 hinge regions. This allows it to adopt many conformations that enhance antigen binding and complement reactivity. There have been older papers that looked at hinge deletions and improved or increased complement activity, as well as a more recent paper looking at association with critical disease and death in the context of COVID. And so because we know that our phenotype is associated with complement, we wanted to look at the presence or absence of hinge deletions in that pilot cohort. So we genotyped our data from IGenotyper. We looked at the presence of either the major allele, here denoted by M, or the shorter allele, here denoted by S, in our cohort. Now, remember, this is only 7 individuals. It's very preliminary. But what we saw was already quite striking. So in our healthy donor cohort, I think 2 people had that deletion, whereas over half of our, so 4 out of our 7 individuals in MG had that deletion in one of their alleles. So this is obviously very suggestive that this could be a genotype that's enriched in individuals that have autoreactive IgG3. In our healthy donor population surveillance projects that we do closely with Corey Watson, we've sequenced over 1,000 individuals. And of those that have been genotyped in the constant region, about 7% of them have this. So we expect that this bar will hold as we expand these cohorts. So, obviously we're rapidly trying to chase down if carriage of this S allele enhances complement activation. So we're doing that both through surveillance at the genomic level of myasthenia gravis patients in this case, as well as some functional genomics in vitro. And we are expanding our cohort to find out. So I can tell you that we have 45 MG patients sequenced and about 35 healthy donor sequenced that are age, sex, and ancestry matched that we're comparing at the moment to expand this data set. But our ambition is to get up to 96 myasthenia gravis patients to really identify how genetics may be affecting the propensity of these individuals to develop autoantibody reactivity. So the last thing I want to leave you on is that this is not just an antibody issue. I know as someone who came for virology to learn immunology that not only do virologists and immunologists not speak at all, but then even with immunology, T-cell immunologists and B-cell immunologists don't speak at all either. And so I do want to make an attempt to get the interest of T-cell immunologists as well. So Oscar also wrote a paper in late 2022 that was published looking at T-cell receptor genotyping as well. And similar to that, seen in IG, there's a huge amount of underappreciated variation in the T-cell receptor loci. So these were 9 unrelated individuals where we found 79 T-cell receptor alpha, beta, and delta alleles. There's also major structural variants. These triangles here represent deletions, and a bunch of the novel alleles are represented by these black dots. So there's so much to learn about these immune receptor loci, so much that we can understand about how variation at the germline impacts effector function and interaction with other molecules. This wasn't surprising to me, right? We all know that HLA is incredibly polymorphic, and if TCR and HLA work like a lock and key mechanism, you would have to have polymorphism on the T cell side to match the polymorphism on the HLA side. So we're also expanding our efforts here, particularly in the context of immunotherapy. So with that, I'd like to wrap up and take some questions. So, I think my biggest take-home message is that immunogenomic variation matters. And I think this is something that is paradigm shifting in the field. It's certainly not something that is taught in those introductory immunology courses. I've given this talk around the world, and I've had people who are the chairs of immunology departments coming up to me and saying, I had no idea that we didn't all have the same genes and they just mix and match differently. I think another thing that I'm incredibly passionate about and that our data demonstrates is that data diversity and representation matter. These patterns appear to be more similar in populations than they do across individuals. And so understanding the population dynamics and the patterns that go with population may help us develop sort of precision therapeutic mechanisms that take advantage of this variation rather than just ignoring it outright. So we've just started to scratch the surface of the extent and the impact of this in our antibody repertoires and our T-cell repertoire. This is not just an antibody issue, so the T-cell people don't get to ignore us. And I think that defining the role that this complexity plays in vaccine-mediated protection and immunotherapy and autoimmunity will suggest whether sort of universal treatment strategies are advisable or possible, and for which diseases they'll be most effective. So we personally are working on a number of these fronts. So when I say we, I don't just mean the Smith Lab. This very much is a collaboration between us and Corey Watson and his team. And so continued population-based immunogenotyping. We have an R24 grant from the NIH to do thousands of individuals over the next 3 years. Again, we're expanding our myasthenia gravis cohort. We're also looking at similar signatures in the context of children who develop acute rheumatic fever after strep infection. And then again, looking at T-cell receptor models in vaccination and immunotherapy. So with that, I'd love to wrap up and take any questions. I want to thank all of our exceptional collaborators. I have a QR code here if you're interested in reading more about the FLAIRR-seq paper. Our teams are amazing. We wouldn't do this without any of the support from our wonderful trainees. So thank you all for your attention, and I'm happy to take any questions.
Wow. Well, that was great.
How to stop sharing what's happening.
Wow, that was great, Melissa, and I'm guessing there'll be questions. I have to kind of scan if people raise their hands. Here we go. Swati has a question. Swati, you're muted.
Oh, she doesn't have a question.
Oh, okay. Well, I have a bunch, but excuse me.
I have a question, if that's all right.
Great.
Hi, Melissa. Awesome talk. Hi. So two questions. So the first is with the FLAIRR-seq. So have you tried FLAIRR-seq with the Connects protocol at all? Or are you still just with the, not trying it?
Yeah, so for those of you that don't know, Connects is the commercial name of a concatemerization protocol that PacBio just released, where because PacBio can do reads up to 20 kb, and our longest antibody transcript is 2, in order to take advantage of the length potential, you actually can then concatenate antibody transcripts to get far more transcripts per molecule, and therefore overall way more depth in your sequencing. So yes, we are absolutely adapting it. That's ongoing work in the laboratory. I think what that will allow us to do is twofold. We can increase the number of samples that we run on the sequencer, or increase the depth depending upon the research question. So at least in the FLAIRR-seq paper, although we generated far less raw reads than, say, a standard AIR-seq 5' race on a MySeq would, after all the filtering and assembly steps that the short read data requires, we actually ended up with more productive reads for AIR-seq analysis post-informatics. And so if we want to keep the depth the same that we're using in, say, the FLAIRR-seq paper, we can increase the number of individuals we have per pool by 3-fold, which of course just brings down the cost. If you have interesting questions where you really want to look for extremely rare clonotypes that may be antigen specific or something like that, then we could just increase the overall depth tremendously. So I think with the standard ISO-seq protocol that Connects is advertised for, they say you can get up to 100 million transcripts per Revio cell. So that would probably be about 150 per FLAIRR-seq Connects, just because the amplicon is smaller. So
smaller. Yeah, that's awesome. I do have one more question. Sorry, that is a follow-up, I promise. So with that in mind, have you used the adaptive loading feature of the Revio at all? And have you tested that with their shorter amplicons? Because I know that's optimized for generally whole genome sequencing stuff. So have you found it's better or worse versus
adaptive loading works at this point across all of the applications. So I would not load the Revio without adaptive loading. So again, for those of you who don't know, adaptive loading is a constant monitoring of the loading of library onto the SMRT Cell during sequencing. Loading for the Revio is optimal around 60 or 70%. And the more that you overload past that point, the less quality your data becomes, because you end up getting multiple molecules per well. And so adaptive loading is an algorithm by which they test the loading capacity every 5 minutes. And when you approach that 60 to 70 point, they cut you off and don't load anymore. So you can really stay in that sweet spot to get the most high quality data out of your run. It's enabled for all applications. I would not load our instrument without it because the cliff of your quality going off, if you overload it all, is so dramatic.
Okay. Awesome to know. Thank you so much.
You get Q60, so we'll let you go on the quality, but
I feel like I just troubleshot William sequencing.
I see Jamie has some questions.
Yeah, I do. Thanks. That was a really wonderful talk and very clear for a pretty complex subject. So thank you for that. Yeah, so I have a couple of questions, and one is that I noticed when you were talking about your long-short allele difference with the deletion in the hinge region. I didn't see any short combinations.
Yeah, I'll have Easton, I know, is on the line. Easton's genotyping right now, that additional 45 individuals. Have you seen a short homozygote, Easton? You want to
I have not, which has kind of been an interesting trend. Sorry, I don't have my camera on. I'm sort of under the weather this week, but yeah, I haven't seen someone being homozygous for that variant allele at all. So yeah,
interesting.
Yeah.
Anyway, so I was just wondering about that, and then also I wanted to ask you about post-translational modification. Because it's kind of important, and and also related to that, whether you think there's any feedback mechanism. So the post-translational modification I'm wondering about is especially with fucosylation versus sialation of the the glycosylation patterns. Because they seem to be related with, inflammatory or not antibodies. And then also in terms of the clonal use of different C regions, constant regions, do you think there's any feedback mechanism for the for the emergence of one versus another, and particularly going into the CH4s, they don't bind to anything, and they also can switch their chains up.
Right.
So I was kind of wondering if you could just kind of whatever you want to say about that.
Sure. So I think the first thing I want to comment on is that there's a comment in the chat from Udalak Jana, who's in Corey's lab, who's done constant region genotyping in 105 individuals. 2 of them have had SS. So he's seen that in 2 individuals.
Well, that's a weird. I wonder why it's so rare, though.
Yeah,
anyway, thank you for that.
That's a great question. So I think where we're starting with the glycosylation question, or with the post-translational modification question, is really identifying those alleles where you gain or lose clear post-translational motifs. So that's most easy in the N-linked motif, right? There's no that I can realize, and I would be happy to be corrected by anyone on this call. As far as I can realize, there's no firmly defined accepted O-linked motif. And then I think, so what we're moving into, hopefully sometime in this year, is to establish a lot of the cell-based assays for functional analysis that others use, ADCC, complement activation, phagocytosis. So because not only have we identified all these alleles with known variation, but we have a ton of unknown variation, like I showed you there with that lysine to asparagine change. And so, we're going to be selecting out variants that appear to be enriched in these disease cohorts with novel variation, and doing what we've all done for years, and cloning them into vectors and seeing what the functions are. I think that you have opened a door to one of my passion projects without even knowing it. So I think that one of the things that's fascinating to me about all of this is the consideration of how we study antibodies, right? When we find a really exciting clonotype, we clone out the VDJ region into an FC vector, right? And yeah. I guarantee we don't even know what allele that FC vector is, let alone if it has the same motif of the FC that's used biologically. And so I think that there's a double whammy there, is the terrifying thought that maybe some of our data is artifactual from the vector we used, but then also the ability to use something like FLAIRR-seq to get that information and then generate the gene string where now you can clone in the physiological full-length transcript and see what the combination of the variable region and the constant region together are capable of doing compared to how we may have done it with previous methods. And that gets a little bit at your question of association with constant regions. It's something that we're very interested in to see if there are specific constant region alleles that are restricted to particular variable region cassettes. We haven't had the numbers or the time to do that analysis, although I think we're getting there as part of
I guess I was more asking about at the protein level.
Yeah, at the protein level.
And there, if you're studying autoimmunity, you can actually get at, you know, some of the antigen specific
and
single antibodies and analyze them.
100%. So I think the other place that takes us to, and you mentioned sort of chain swapping, is in the IgG4, right? So this duplication that we see, which is quite frequent, and the idea that we know that IgG does a chain swapping event, they call it Fab exchange, but it's really the whole heavy chain. That keeps it from being able to cross-link. You know, what is happening in these individuals that have more than 2 alleles, that have 3 alleles or 4 alleles, and does that enhance overall chain swapping? How does that impact susceptibility to that sort of tempering effect of IgG4 populations and chronic inflammation? I think there's so much to be discovered. It's all just really exciting. I have no idea about the feedback loops. I'm not even going to try to attempt that question. But thanks for your question. It was great. It brought up a lot of things I like talking about, obviously.
Yeah, a lot of good questions. I don't see any more right now. Maybe we'll take that opportunity then to get a heat up coffee break